Hello everyone. So today we'll be doing a short mini lesson and we're going to be learning about the absorption and the action spectrum of light, and then we're also going to do an application problem so that we can see, how we would apply these concepts. Now this comes from unit 3, specifically photosynthesis. So because of photosynthesis and the absorption of light energy, we need to have a bit of an understanding of the electromagnetic spectrum as well as the action and the absorption spectrum. This is a very common AP exam question. We're going to look at a AP exam question from just a couple years ago that was looking at the absorption of different wavelengths of light, and they did it in a long answer FRQ style question, which students, tell me they have the most trouble with. So we're going to go through the lesson. I'll teach you everything that you need to know for action and absorption spectrums, and then we're going to do that practice question. Okay, so these first couple slides we're just going to be, explaining absorption of sunlight and then the action absorption spectrum, and then we will go on to our practice question. Now to start off, about the absorption of sunlight. So for photosynthesis, we looked at how light energy that comes from the sun is able to be converted. Now that first needs to be absorbed. So light has to be absorbed, and that's going to be all dependent on, the wavelength of light and how effective a certain pigment is at absorbing that wavelength of light, and that's ultimately going to affect photosynthesis. So light energy is a form of electromagnetic energy, and the distance between crests of electromagnetic waves are called wavelengths. So the crest of a wavelength is the top part, so the very highest point. So if we were to look over here at these red ones, the crest of this one would be here, and the crest of this one would be here. It's the very top point. Now the wavelength is the distance between those crests. That's the length of one wave, because then this would be the next wave to the next crest, and so on and so forth. So that determines how long a wavelength is. Now we can see that these ones are pretty long over here in red, but if we look over here, towards this blue end, these ones are quite short. The distance between them is very short, meaning they have shorter wavelengths. Now this entire range of radiation is known as the electromagnetic spectrum. So the entire range of wavelengths that are possible is what makes up our electromagnetic spectrum. So our really long wavelengths over here would be radio waves, and then you go into infrared, and then the smallest wavelengths would be our gamma rays over here. Now the only wavelengths that are important to us are the ones that fall between this really small range right here. So between here and here. Now that's the visible light spectrum. So despite all of these wavelengths existing, we can't see X-rays, we can't see gamma rays, we can't see radio waves, but what we can see is just between these 2 points here, which is the visible light spectrum. Now this is a very narrow band, and it's between 380 nanometers and 750 nanometers. So that's the wavelength. So the wavelength is between 380 nanometers as a measure of length and up to 750 nanometers. Now that's what's known as the visible light spectrum because it's detected by the human eye. Now we're going to look at light as though it consists of discrete particles called photons. So when we talk about the absorption, we really just talk about, a photon, a single kind of unit of light striking down and hitting, a pigment molecule for absorption. It's not going to be a stream of light. We're going to think of it as one discrete particle. Now, the amount of energy is actually inversely related to the wavelength of light. The shorter the wavelength, the greater the energy of each photon. So, the longer the wavelength, the smaller the amount of energy. So, the ones over here with really long wavelengths, so radio waves are actually the lowest. So, they have the lowest frequency, so lowest energy. And these ones over here that have, are very close to one another, these have the highest energy. So radio waves are the lowest, and gamma rays, for example, are the highest. Now going on to pigments, the substances that absorb visible light are what's known as pigments. Now those pigments are going to be found, in our chlorophyll molecules, which are absorbing the light energy from the sun into the plant's chloroplast. Now these pigments are what are actually absorbing that light. And you've probably heard of the word pigment in terms of colors and things like that. But these are actual molecules within the membrane of our thylakoids that will be absorbing the actual visible light from the sun. Now different pigments absorb light of different wavelengths. So each pigment is very good at absorbing only certain wavelengths. Nothing is good at absorbing all the wavelengths. They're very specific. So When we see a green plant, which is going to be most of our plants, they are green because chlorophyll absorbs violet, blue, and red light while transmitting and reflecting green light. Now, whenever you look at something, that's because it's reflecting that color into your eye. Now, that means that they're absorbing every other color in the visible light spectrum, and the only one that isn't being absorbed is being reflected, and that's what you're viewing. So for a green leaf, for example, that leaf is reflecting green, therefore it's absorbing purple, blue, red, and orange. So if we look at chlorophyll, we can look at specifically chlorophyll A, which is this light green line here. It's really good at absorbing purple and blue light, but it's not very good at absorbing green light because it's actually reflecting green light. And then it's going to be good at absorbing orange and red. It's also going to be good here, purple and blue. So it's going to absorb all the other wavelengths, and therefore that's why it reflects green. And so that goes for anything. Whatever they're really good at absorbing, they're going to reflect the opposite color because they're not absorbing it. So that's going to be reflected, and that's what you're going to see in your eye. So if we look at this graph here, this is a graph plotting a pigment light absorption versus wavelength, and it's called the absorption spectrum. Now, there's the absorption spectrum and there's going to be the action spectrum, so we're going to look at both. This one here is the absorption spectrum. So it is a graph plotting a pigment's light absorption versus wavelength. So it's wavelength of light, and then the y-axis is amount of light absorbed. Whenever you have a graph and you're trying to figure out what it is, always look at your x and y axis. That's going to be the biggest indicator, of what they're trying to tell you and what they're trying to show you. So this, if you can see, amount of light versus wavelength in nanometers, and they give you the color at each wavelength, so you can also see, what color is it best at absorbing, as well as the actual length of the radio wave. And then the amount of light absorbed here is going to be how much is being taken in and absorbed by each one of these pigments. So carotenoids, we have chlorophyll A and chlorophyll B as, 3 separate ones. So this is showing you the entire absorption spectrum for all 3 of these pigments. We're going to see that the action spectrum only shows you one line. Since there are more than one type of pigment molecule within a plant leaf, they're going to be good at absorbing different wavelengths. Now that's going to be helpful because you're able to absorb a wider range of wavelengths. So for example, chlorophyll A is, really good at absorbing this pink one at around, 440. However, carotenoids are good at absorbing over here around the blue spectrum around 490. So you get to cover a wider range of absorption, and the more absorption you get, the more photosynthesis you get. So by having this sort of range where, they're not exactly over top of one another, each pigment is kind of good at absorbing a different wavelength, that's going to give you a wider range of light that you absorb into your plant cell, and that's going to give you a higher rate of photosynthesis. So there are 3 types of pigments in chloroplasts. You have chlorophyll A, you have chlorophyll B, and you have carotenoids. Chlorophyll A is the key light capturing pigment that participates directly in the light reactions. So chlorophyll A is going to be the main one. It's going to do majority of the absorbing, but it's not the only one. So you're going to get a lot of absorption where chlorophyll A is, but you're also going to get, other wavelengths being absorbed by your accessory pigments, which include chlorophyll B. So chlorophyll B is this green one over here. Now this one is a little bit shifted over, a little bit better at absorbing blue light, and then you're going to have your carotenoids, which are a separate group of accessory pigments, and they absorb violet and blue-green, and they emit shades of yellow and orange. So the carotenoids are actually what make up carrots, and so you can remember that because, they are reflecting yellow and orange light. So if you look at where the carotenoids end, you're going here, and they're not doing any of this yellow, orange, and red light. That means they're reflecting it, hence why carotenoids show up as orange. However, they are absorbing everything else, which would be, this purple, blue, and, green sort of spectrum over here. So the accessory pigments are going to function to broaden the spectrum of colors that drive photosynthesis. Okay, so you get a little bit of a wider range, like we said, in terms of the wavelengths that are being absorbed. Now, you also have something called the action spectrum. Now, this is the absorption spectrum, and this is the action spectrum. You have to be able to tell them apart as well as explain the differences between them. So typically the absorption spectrum, if they're going to show it for an entire chloroplast, then you're going to have more than one line. The action spectrum is only going to be one line. The y-axis for the absorption spectrum is going to be absorption of light by individual pigments. So it's going to be absorption of light per pigment. Then the action spectrum is looking at the rate of photosynthesis. So obviously the absorption of light is related to the rate of photosynthesis. The rate of photosynthesis is just kind of showing you the overall effect of the absorption at all of these wavelengths. And then the x-axis is the same for both of them. It's just going to be the wavelength of light. And if you look at it, the graphs are pretty similar. The absorption spectrum, if you kind of put all of the lines together and kind of standardize them or made them an average, so you would get the action spectrum. So you can see that, you have a good amount of absorption in this purple and blue area. You get little to no absorption in this yellow and green area. And then you get a big jump of absorption around the orange and red. And so that is exactly what's happening here, because as you absorb light, you're also increasing the amount of photosynthesis. You're having more light energy available to be converted. So the action spectrum is looking at the actual action of what's happening in this process. So the rate of photosynthesis, usually it's measured in the amount of O2 released, because O2 is a byproduct. So most of the times you can measure the rate of photosynthesis by looking at how much O2 is released, because that's the end product. So again, just to recap, the absorption spectrum is going to look at the absorption of wavelengths per individual pigment and how much light they actually absorb. And then the action spectrum is showing you the rate of photosynthesis at each wavelength. Now they're related to one another, but they don't show the same thing. This one's showing you rate of photosynthesis and the actual action of this set of steps. However, it does relate to the absorption spectrum in the sense that, as you increase light absorption, you're also increasing the amount of photosynthesis that you're doing because you're just getting more reactants to then turn into products. So now that we've talked about that, I'm going to move on to going to an application of this, and this is actually going to be a mock AP exam question. So this was on the AP exam, a couple years ago, and this was looking at spectrums in terms of light. So let's read this question first, and then we'll look at the graphs. So this says, an absorption spectrum indicates the relative amount of light absorbed across a range of wavelengths. The graph above represents the absorption spectrum of individual pigments isolated from two different organisms. One of the pigments is chlorophyll A, commonly found in green plants. The other is Bacteriorhodopsin, commonly found in photosynthetic purple bacteria. The table above shows the approximate range of wavelengths of different colors in visible light spectrum. So that's a lot to kind of get through. So we're going to look at, just the important bits, and we're going to highlight it. This is a relatively short question compared to the ones that other ones do in terms of, sometimes the question is a couple paragraphs long. So for this one, we're still going to go through and highlight, the important things that we need, just to make sure that we're keeping them in mind and practicing those skills for, ones where they're a little bit longer as well. So we'll just get that practice in. So it says the graphs above show absorption spectrum of individual pigments. So that's important. We just talked about the difference between absorption and action spectrum. So we're not looking at the rate of photosynthesis, we're looking at the absorption per pigment. Now, there's two graphs here, and they're saying one of the pigments is chlorophyll A, which is commonly found in green plants. So chlorophyll A of green plants, and the other pigment is Bacteriorhodopsin, which is commonly found in purple photosynthetic bacteria. And then we also have this graph or this table that shows the ranges of wavelengths of different colors. Okay, so for this, this is one of those tricky ones that students might get a little bit overwhelmed with, especially because in terms of talking about pigments, we only really talk about chlorophyll A, like what I talked about in the past slides, or everything you need to know for action and absorption spectrum. However, you can see here that the AP exam is asking you something about Bacteriorhodopsin, which is another type of pigment that we haven't covered. Now, you don't need to have any background in Bacteriorhodopsin. This is kind of where the fact that it's an application question comes into play, because you're going to use your knowledge of action and absorption spectrums and pigment molecules and apply it to a new, pigment molecule that you don't really know anything about, such as Bacteriorhodopsin. So we're going to practice that. Now, if we look at A says identify the pigment, whether it's chlorophyll A or Bacteriorhodopsin, used to generate the absorption spectrum in each of the graphs above. Explain and justify your answer. So they've highlighted, the key terms here, identify, and then you have to explain and justify for both of them. So this one is likely going to be worth 4 marks. Now you're identifying the pigment used to generate each one of the graphs. So you have graph 1 here, and you have graph 2 here. So one of these it's saying is chlorophyll A, and one of these is bacteriorhodopsin, and you have to guess, which one is which and explain why. Now, that seems quite difficult because we're not given a lot of information, especially because, we don't know anything about bacteriorhodopsin. However, we do know what color each one of these pigments reflects. Now, that's the key part here, and this is why I highlighted. So chlorophyll A is green. So if we write here, chlorophyll is green, and bacteriorhodopsin is found in purple bacteria. So bacteriorhodopsin is purple. So that's all we need to know to answer this question. So they're asking, which one of these represents the absorption spectrum of a pigment that is reflecting green and a pigment that's reflecting purple. All you need to know for bacteriorhodopsin to be able to match it to one of these graphs is, what it absorbs or what it reflects. And then you're going to be able to see on that absorption spectrum. So because they are talking about the actual color of the pigments, they're not talking about, the actual colors that they're absorbing. They're looking at the colors that they are. Therefore, we're looking at the colors they're reflecting. So chlorophyll A reflects green light. That means it's going to absorb every other color and it's going to not absorb green light. If you're reflecting green light, that means you're not absorbing it. You're absorbing everything else. So if we look in our table now, we need to be able to use this table. If you go through a whole question and you haven't used one of the tables or one of the graphs, that's probably a red flag that you're missing something. So again, you would have to use this table and make sure that you use it while you're answering the question. So chlorophyll is green, and the green spectrum goes from 495 to 570. So let's point that out on our graph. So wavelength is down here. Let's mark those out on our graph. So 495 to 570. 495 to around 570. 495 to 570 around here. So these ones is the green wavelength. So for chlorophyll A, it absorbs all the other colors and it reflects green light. So it should not absorb green light. So chlorophyll A is green. It should not absorb any green light because it is reflecting green light. Now, if we look, our y-axis is relative absorption. So if we need to figure out between graph 1 and graph 2, which one is the green one, it's going to be the one that doesn't absorb any green light. This is our green light area. Then clearly, this one is going to be graph 2 because there's very minimal absorption of light that's green. That means it's going to reflect as a green color and likely be chlorophyll A. In graph 1, you can see that, there is a good amount of absorption of green light. Hence, that pigment would never show up as green. It'll only show up as green, if you're actually reflecting and not absorbing any green light. So this one would likely be chlorophyll A. Okay, so now we're gonna write out the full answer so that we can make sure that, we get all the marks. So just saying that, graph 2 is chlorophyll A is not enough. You have to identify and then explain and justify your answer. So let's put this on the next page. So we'll write here that, for A, graph 2 is chlorophyll A. Since it is green, it should reflect the green wavelength, which is from 495 to 570. And then therefore low absorbance at that wavelength. You could also say that, that means it absorbs blue and purple, and it also absorbs, absorbs red and orange. So it should be absorbing those, hence why, you have a large absorption here and a large absorption there. You could use either way. It explains the same thing. Just for time's sake, I'm just going to write down a little shorthand for it. You should always write full sentences on your exam. You don't have to write too much as long as, it's a full and complete sentence. So the key for this is just process of elimination. You're going to make sure that this one aligns with your explanations as well. So this one would have to be, bacteriorhodopsin, but you always want to check. You do always want to check, because you can't, always trust that your first one was done correctly. If you can be confident that both of them work with your explanation, then you can be sure, that your labeling has worked. So it's commonly found in purple photosynthetic bacteria. So if it's purple, it should be not absorbing any purple wavelengths, and it should be reflecting purple, and it should absorb all the other wavelengths. So it should have high absorption, between here and here, and low absorption between these two wavelengths. If we look at our graph for graph 1, which we're assuming is bacteriorhodopsin, we can see that for the violet and blue wavelength, so from about 380 up until, around 475, there is low absorption. There's still a little bit of absorption, but it's still a lot lower than all the other ones. Therefore, that's the color that it's going to reflect. So it's lowest here, and then you have high absorption, between 500 and 700. Hence why there's a large amount of absorption between there. So this one would clearly be, bacteriorhodopsin, because there is low absorption of purple wavelengths, so between 380 and 475, and a high absorption of all the other ones, meaning that those are absorbed and the color that's reflected is purple. Because bacteriorhodopsin is a purple pigment, that has to be the absorption spectrum for it. Okay, so then you would have to say graph 2 is chlorophyll A, graph 1 is bacteriorhodopsin. It's purple. Again, write full sentences, but just for the sake of time, I'm just going to write down the main points. It's purple, so it shouldn't absorb purple, which goes from, I believe, around 400, purple and blue, between 400 and 500, just as a rough estimate. And there was graph 1 showed low absorption there. Okay, so that is it for the first part. So that should get us about 4 marks. Now we're going to go on to B. And B says, in experiment, identical organisms containing the pigment from graph 2 as the predominantly light-capturing pigment are separated into 3 groups. The organisms in each group are illuminated with light of a single wavelength. The first one has 650 nanometers, the second one has 550 nanometers, and the third one is 430 nanometers. The 3 light sources are of equal intensity, and all the organisms are illuminated for equal lengths of time. Now the question is to predict the relative rate of photosynthesis in each of the 3 groups, and then justify your predictions. Okay, so that was quite a long one. Let's go through and erase all the markings that I have here. So for this, it's asking you to only use graph 2. So we're only using the one for chlorophyll. So it's saying that you had organisms that were related with a single wavelength. So you had 650, 550, and 430 were the 3 groups. 650, 550, and 430. Okay, those are your 3 groups. And then they said that they did it at the same lengths of time for the same intensity. So that's just to standardize it all. So the only thing basically that's the difference between these 3 groups is the wavelength. They're getting the same amount of light for the same amount of time. They're just at different wavelengths. So we're going to see how wavelength affects that. And then it's going to say predict the relative rate of photosynthesis in each of the 3 groups and justify it. So we'll use graph 2, we'll look at these wavelengths, and then we need to be able to tell from this graph, what the photosynthesis rate would be. So let's start off with 650. All you have to do is, look at where it lies on the graph. So 650 is about here. So our point of absorption would be around here. So not the highest amount of absorption, but you get a little bit of absorption at 650. Now let's look at 550, which is here. So you're getting virtually no absorption of light. And then at 430, which should be, around here, you're actually getting the most amount of absorption. So in that case, we can now see that, 430 has the highest amount of absorption and 550 has the lowest amount of absorption. So what does that mean for their photosynthesis rate? Which one is going to have the highest rate of photosynthesis? Which one is going to have the lowest? Now, based on what we talked about for the absorption versus the action spectrum, we remember that, the absorption is correlated to the actual rate of photosynthesis. The rule was the more light you absorb, the more photosynthesis that's going to happen, or the faster the photosynthesis is. Because the more light you have, the more you're able to do photosynthesis and convert that to your products. So therefore, we can assume that, the one that absorbs the most amount of light will have, the highest rate of photosynthesis. So which one, has the highest absorption? That one's going to be at the wavelength of 430. So at 430 nanometers, chlorophyll A is going to have a really high level of photosynthesis because it has a really high absorbance. So 430 is going to be the highest. 550 is going to be the lowest. And 650 is not incredibly high, but it's higher than 550, so we'll put this as the intermediate. So highest amount of photosynthesis would be at 430, and lowest at 550, and 650 is an intermediate. So it says predict the relative rates of photosynthesis in each of the three groups and justify your prediction. So for this one, I write out the entire thing. You would have to say for each one of them. So just saying that 550 is the lowest and 430 is the highest is not enough. You have to say all three of them. Describe their rate. Students might get confused because you think about rate and you think that you have to actually calculate it. These are pretty relative, so it's just going to be, this one's the highest, this one's the lowest. You're not actually going to get, like a numerical output. And then for the reason why, you can say it goes the same for all three of them. So you can say, 430 is the highest because it had the highest absorption. More absorption of light means increased rate of photosynthesis. And then for 550 and 650 as well, the same rules apply. 550, you can say there's little to no absorption of light at the wavelength of 550 nanometers. Therefore, you're going to get really low photosynthesis because, the less absorption of sunlight, the less photosynthesis that would be running. So that is B. We're not going to do C for this one because this one is asking about an evolutionary history. And so evolution is, I believe, Unit 8. So like I've mentioned before, the FRQ questions on the AP exam are going to, put together a bunch of different concepts from each one of the chapters. So here you can see chapter 8 as well as chapter 3. So you're going to have to be able to make those connections. Because I haven't gone over chapter 8 with my class, we didn't go through C. But when we go through all the chapters, obviously we'll come back to this question and do part C. Now, the last thing that I want to do is to look at, the practice answers. So for this, we're going to look at the actual marking scheme. Now this, I always say, is very, very helpful for students because you can see how the AP examiners are going to be marking your test. Now that's really important to see, what they're specifically looking for, how they want you to organize it. And if you practice enough of them and you look at the answer guides as well, you're going to get pretty used to the level of specificity and the level of depth that they're looking for. A lot of the times you'll be quite surprised with the amount of actual content that they're asking for you. So for this, we're going to go on to the one for part A. So part A was identifying which pigment was which and then explaining why. So this is the answer guide for this one. So you had to identify both of the pigments and then you get 1 point each for explanation. So we said that graph 1 was Bacteriorhodopsin and graph 2 was chlorophyll A, so that is 1 mark. We had to explain that an organism containing Bacteriorhodopsin appears purple because the pigment absorbs light in the green range in the light spectrum, so you could have said or. So like I said, you can either say, it absorbs green light or that it reflects purple light and something like that. So you could use either one, they indicate the same thing. So we more so focused on the reflecting, so we said this part, and you could have gotten the full mark for that as well. So we said that that's because it reflects purple or red-blue light, and the reflected red and blue light appears purple. Okay, so we know that graph 1 is Bacteriorhodopsin because there is a low absorption of violet light, and therefore that's why the color is purple. So that would have gave us 1 mark as well. And then for the second one, for chlorophyll A, you could say that an organism containing chlorophyll A appears green because the pigment absorbs red and blue light, or you can say that it appears green because it reflects green light, which is what we mentioned. If you did either of them, you would have gotten the mark. They accept both because both are technically correct, but you don't have to mention both. You only need to mention one for the mark. So this would have gave us 3 out of 3 for something quite simple. So not a lot of explanation. All you really had to say, for example, is chlorophyll A appears green because, it reflects green light, low absorption of green on graph 2. Okay. Now for part B, this is how they wanted to do it. So it was 5 marks. You get 1 point for determining each one. So that 650 was intermediate, 550 was lowest, and 430 was highest. So we would have gotten the marks for that one. So 1 point for each, and then you could have said for 1 point each in each one of these boxes, you had to explain why. Now the explanation for why is the same for all of them. So we can look at the highest one first. So the highest absorption occurs at 430, therefore the greatest amount of energy is available to drive photosynthesis. So like I said, more absorption of light means more energy and more reactants available for photosynthesis. It increases rate of photosynthesis. Same thing for 550. It's the lowest rate, therefore there's the least amount of energy that's able to do photosynthesis. So you're going to have a lower rate of photosynthesis. So we would have got, maybe they meant that this one was out of 6, but either way, we got all of these. And this little note at the bottom just says that, students that combine 650 and 430, so saying that, these have similar rates, you would not accept it. So it's just saying that, it's not enough to say these are the highest, these are the lowest. You had to mention all 3 of the groups. So say that there was a highest, a lowest, and an intermediate to get all 3 of those marks in this category here. Okay, so I just talked you through, how you're going to answer these AP exam style questions, as well as taught you the entire lesson for it. So now you guys are fully prepped for any action and absorption style questions. And now we kind of get a good sense of the concepts that we're going to learn and, how we're going to apply them. And so once I explain it, very thoroughly and go through the content entirely, we can see that, these AP exam and application questions aren't too scary. It's just the way you think about it and making sure that, you have good organized notes and you're fully understanding the questions, as well as, practicing the techniques that we're talking about in terms of how to approach application questions. Okay, so that's the end of this video, and then I'll see you on the next one.